All right, and today we're going to be discussing the topic which immediately follows lipids, which is membranes and membrane transport. And I think this is a really interesting lecture because the way membranes are classically viewed is as compositions of lipids which provide a barrier between the intracellular and extracellular environments. But what we're going to do with this lecture is really elaborate on the role of the membrane and the composition of the membrane and the way the membrane is used to facilitate homeostasis and metabolism. So with that said, I'm going to have to move a little bit on the fast side because, well, we have a lot of content to cover. So the general outline for this lecture is what are the chemical and physical properties of the membrane? What are the structures of the membrane and its proteins? How are these membranes organized, and what are the dynamic processes which modulate their function? How does transport occur? And specifically get into the details surrounding passive diffusion, facilitated diffusion, and active transport. And these last two I've actually edited out except for a few examples of ion gradients because the volume of information is just a little prohibitive for a single lecture, and the content in the book is really adequate for fleshing out some of these other topics. There's nothing really conceptually abstract that you can't just get from looking at the figures and reading that content. So, membranes. What are the rules? Well, we've talked about membranes as a barrier, like I said, and we typically think of membranes as bilayers of lipids which form semi-spontaneously, which are one of the foundation elements of life because they keep the internal environment of the cell separate from the external environment. But as part of that, there's a lot of secondary functions other than just maintaining a barrier. So yes, they're barrier to toxic compounds, but they also are essential for regulating transport and accumulation of nutrients. They're essential for energy transduction. They facilitate cell motion. They're actually very dynamic, and we're going to look at how vesicles form and are absorbed a little bit. They're essential for reproduction because of the same facilitation of motion in the membrane, ability to divide. They're essential for signal transduction, and they're essential for cell-cell interactions. And those last two are really, really important because the membrane and the proteins which are integral in the membrane are crucial for communicating or receiving information from inside the cell to outside the cell. They're also essential for receiving information from the extracellular environment and communicating it to the inside of the cell so it can respond. And in cell-cell interaction, you see that contact between cells often regulates the expansion of cells or the metabolism of a cell or the way the cell is primed to react to its environment. So particularly in immune function. So when we're talking cell-cell interaction, often we can think about immune function as well. So one of the things we want to dispel right out the gate is that cells are their membranes are entirely comprised of lipids. Yes, lipids play an incredibly important role. They're the foundation of the membrane. That's why they're called lipid membranes. However, these membranes have a very high content of proteins. The protein content is variable from membrane to membrane, tissue to tissue. But a biological membrane can contain up to 80% protein by mass, or as little as 15% protein by mass. So some sections of membranes, like in neurons, will be very low in protein, whereas others, membranes associated with mitochondria, will be incredibly high in protein. And that is, like I said, variable. One of the ways you can think of when you have something that's 80% protein, there's just enough membrane to glue the proteins together in these multi-protein constructs where you have one protein after another that are sequenced spatially so that a substrate can go from one protein, and that product can then become the substrate to a second protein, and then that product can become the substrate for a third protein in a very efficient sequence of chemical reactions that are facilitated spatially by the membrane. And these membranes, like I said, full of proteins, carry out all kinds of enzyme catalyzed reactions and transport activities. So whenever you have a biological function which is really facilitating chemical reaction and transport, you're inherently going to have an expectation that it will have a higher percent protein composition. And now, let's see here. Just below, we can take a look at some of the variability in lipid composition of different membranes. And you can see we have the key on the left side with the different categories of lipids, including phosphatidylcholine, phosphatidylethanolamine, sphingolipids, phosphatidylinositol, etc., going down towards cholesterol. And on the far right, we have the different subcategories of membranes. We've got a nuclear membrane, Golgi, mitochondria, lysosome, and plasma. And these are arranged in a decreasing level of phosphatidylcholine from the nuclear membrane being the highest at almost 50% to the plasma membrane, which has 28% phosphatidylcholine composition. But all these are different subcategories of phospholipids, minus the cholesterol and cardiolipin and minor lipid. But you can see that different components of cells and different tissues will have substantially different lipid compositions. And these lipid compositions are finely tuned to facilitate a specific biological function. Now, one thing this book doesn't really talk about, but I want to stress here, is that you can think of the lipid membrane as a tunable, two-dimensional solvent. And this is really essential for figuring out how a membrane functions. So by tuning the lipid composition, you're tuning the specific chemistry of your solvent. If you need, say, an OChem to do thin layer chromatography, you can perform minute changes to the ratios of your polar to nonpolar solvent and maybe acidify that solvent combination so that you're finely tuning the way different combinations of eluents migrate through the silica gel. Now, in the case of the membrane, the lipid composition is that finely tuned solvent. So by changing the ratios of these different lipids, they're changing the chemical characteristics of the membrane, and they're changing the biophysical characteristics of that membrane so that the environment is perfectly suited for different proteins to function elegantly when housed within those membranes. Another really, really important component of this statement is it's a two-dimensional solvent. By placing all of these proteins associated with regulation and communication and transport at the membrane, you get the ability to localize them next to one another very efficiently. If you try to get two different things to coordinate with one another in 3 dimensions, that's fairly complex. To get several things to coordinate in 2 dimensions is a much easier task biologically and in general. So what we have within the membrane is the ability to coordinate a lot of biochemical reactions very efficiently and to tune those biochemical reactions very effectively by changing the lipid composition regionally around them. So that's my pitch for the complexities of membrane biochemistry. And moving down the list here, what are the chemical and physical properties of membranes? Well, this is the simple version. We're taking a big step back from what I was just talking about, and you can see that very few lipids exist as monomers. We've talked about that. So what happens is, to reduce the magnitude of clathrate production in the solvent and to maximize entropy of the system, they organize so that the lipophilic tails or hydrophobic tails interact with one another or hide from the solvent, and the polar groups face outward towards the solvent where they can react favorably. And this can result in a couple different structures. You can have micellar structures, you can have vesicular structures, but we typically think of lipid membranes as the bilayer structure. And I really like this picture of the bilayer because most bilayers, you see this really rigid form of the lipid tail where they line up perfectly and straight. And this one in the bottom left corner really shows how flexible those tails are. Now remember, the lipid components, the fatty acid chain components of these phospholipids are highly flexible. They require space, they're mobile, they slide around one another, they move in and out of each other a little bit. So you get a lot of van der Waals interactions, and you get the ability to compress because they can slide into and over one another, and expand because there's nothing really to say that there can't be a slight void in there as well. Also, you have variability with the thickness of the membrane because the length of the carbon chains in the lipid components can vary. They can be 12, they can be 24. I mean, they're not typically 12 or 24, they're usually in the 16 to 18 range, but there's variability there. So you can have variability in the thickness of the membrane as well. Now, there's a chemical term called the critical micellar concentration, which defines the amount of lipid which must be dissolved to spontaneously form a membrane type structure or bilayer structure or micellar structure. And this is variable from lipid subtype to lipid subtype. It's one of the biophysical characteristics which is unique to each lipid. And it's also not necessarily specific to lipids. It turns out that we make a lot of micellar structures out of non-lipid compounds which have polar ends and nonpolar ends and have the same phenomenon as the lipids do, forming micelles. So as in grad school, I worked with highly modified cyclodextrin compounds which had incredibly low critical micellar concentrations. They would form micelles in aqueous solutions at very low concentrations. It turns out, even though this is a spontaneous event for lipids, it takes a reasonable concentration of lipids to spontaneously form bilayers. But it still works in biological systems, no problem. Obviously, as there's tons of evidence of that working. So I talked about the combination of lipids and proteins and how lipid composition is highly variable, as well as there's a rationale for having these proteins accumulate on a two-dimensional surface for efficiency of organization and regulation, as well as just being essential for communicating intercellular and extracellular events and regulating transport so that you can have control over the cellular environment. Now, the model which describes this, which really gets you away from the visualization of the lipid bilayer, is called the fluid mosaic model. And it was really proposed, a while ago, like 48 years ago, by Singer. And the model is just a proposition that the phospholipid bilayer is a fluid matrix. So there's a lot of mobility here. You can see that the proteins are set into the membrane, but they're not locked into a single location in the membrane. So they can slide around in these two dimensions from side to side within the constraints of the cytoskeletal structures which are integrated into the membrane as well. And this allows for regulation where they can interact with each other within the two dimensions of the membrane or be separated. Things can be recruited to them from the cytosol to change the way they are functioning or regulated. And within this model of the fluid mosaic model, there are two general classes of proteins. There's the peripheral protein and the integral protein. And the peripheral proteins are ones that associate with it, the membrane, on the surface, through weak interactions typically. And you have the integral proteins, which are physically integrated into the membrane. And they don't necessarily often span the membrane, but they frequently do. So I've said this, I get ahead of myself sometimes in lectures, but there's a lot of motion in the bilayer. Lipids, tails can bend and tilt and rotate and slide around each other. They're very dynamic, particularly under biological temperatures. And they lie typically perpendicular to the surface, but they don't exist rigidly perpendicular to surface. They can angle off, they can need some elbow room, particularly if they have that cis double bond or a few cis double bonds in them. They're going to need a little bit of wiggle room, and that's going to cause a lot of motion around that zone of that single lipid, which will cause a lot more membrane fluidity because there will be less packing. But not only can proteins migrate, lipids themselves migrate. They're not covalently linked to one another. So a lipid can move from, any single lipid can move around quite a bit within a membranous structure. So let's take a little bit at integral and peripheral proteins, and we're also going to look at a subcategory of this, lipid-anchored proteins. If we're talking right about peripheral membrane proteins, peripheral membrane proteins are proteins which associate with the membrane, but they're not strongly bound to the membrane. They're not held within the body of the membrane. And they can associate via ionic interactions and hydrogen bonding with the polar lipid head groups, or they can interact with a nonpolar membrane core by inserting a hydrophobic loop just underneath the polar head groups of the lipids so that they're pushing this helical structure down into the hydrophobic region of the membrane, and this causes a nice interaction which resists the lipid from separating from, or the protein from separating from the lipids by virtue of this strong nonpolar interaction. So one of the things that you can do experimentally in the laboratory is you can easily dissociate most of these peripheral membrane proteins from the membrane through mild detergent treatments or aqueous treatments with some salt solutions, which break up these ionic interactions or hydrogen bonding interactions. And that's how you can easily separate a membrane from the cellular tissue, and then you can segregate the peripheral proteins from the integral proteins, because integral proteins, which are spanning or physically integrated into the membrane, aren't going to easily separate by virtue of a mild detergent. You have to physically break the membrane apart through a more aggressive solvent to pull those structures out. Now, if you look at the bottom right corner, you can see that there's another category of peripheral membrane protein, and this type of peripheral membrane protein associates with an integral protein versus the surface of the lipids themselves. And that's just another variation on the theme. So as I mentioned just in the last slide, integral membrane proteins are firmly anchored. They're resistant to detergent treatments. It's really common, by the way, for those of you who are prepping for the MCAT, to see a question regarding that specific question. What if a protein's detergent resistant versus non-detergent resistant, and what subcategory it may be. And just pay attention to that because it's a really common theme in some of the multiple choice questions. So the integral proteins are strongly embedded in the bilayer, and they don't always span the whole membrane, but they very often do. And not only do they often span the membrane, it's really common for multiple sections of the protein to weave in and out of the membrane multiple times, which we'll take a look at in the next slide. But let's see here. These proteins are resistant to your salt washes and your detergent treatments, unless you use very strong detergent, which can physically break the membrane apart. So, like I said before, it's very common for there to be multiple transmembrane domains. And whereas I'm going to go back to the previous slide, if you look at the images below here in A and B, you can see you have these helical structures which span from one side of the membrane to the other. And this is what's called a transmembrane domain. So just as we've discussed helical structures that bury themselves at the center of the protein because they're covered with hydrophobic amino acid residues, these transmembrane domains have to have non-polar amino acid residues to slide easily and securely fit and be happy within the membranous environment. So what you see is you have regions of polar R groups for the amino acids on one side, on the outside of the membrane, where it's interacting with your aqueous environment, and then within the membrane you have sequential regions of nonpolar amino acids. And this gives you these transmembrane domains. Now, whereas in the picture that we just came off of had a single transmembrane domain, it's much more common for integral proteins to have multiple transmembrane domains, and it's common for to have anywhere between 2 and 12 transmembrane segments. Most transport proteins, because you're dealing with making a pore within a membrane that can be regulated, are going to have anywhere between 6 and 12. And sometimes you have a lot more, but that's a lot less frequent. So this is what it looks like. This is a classic example of a integral membrane protein, which is also a pore or pump. This happens to be Bacteriorhodopsin, and it's a light-driven proton pump. And what this consists of is 7 different transmembrane helical segments, which have very short loops connecting them. So there isn't a lot of extra protein hanging out in the aqueous environment. It's very efficient at connecting one end to another from one section of the domain to the other. And the 7 different helical structures orient themselves relative to one another to form a pore/pump, which can interact with photons to provide the energy to actually pump protons from one side of the pump or pore to the other. Now, I was talking about regions of hydrophobic versus hydrophilic R groups on amino acids if you were looking at the primary sequence. And when we do this, it's one of the ways we can predict what proteins exist within the transmembrane sections of a membranous protein. So these plots are called hydropathy plots, and each amino acid has a different hydropathy index, meaning how hydrophobic is it. And when you have, if you're looking at this hydropathic plot in the bottom right corner, you can see distinct regions 1, 2, 3, 4, 5, 6, 7 of the this particular protein, which have groups of non-polar amino acids, followed by very short segments of polar amino acids, which are just those connecting loops. So if you're just looking at primary structure of a protein, and you see blocks of non-polar amino acids, and then you see very short segments of polar amino acids connecting those blocks of non-polar amino acids, you can take a pretty educated guess that you're dealing with a transmembrane protein. Now, another clue would be that the number of amino acids to span that section are going to be fairly similar. There's not going to be fairly low variability in the number of amino acids per block, because they're usually going to efficiently transect a membrane, which has a little bit of variability in thickness, but not necessarily a high degree of variability in thickness. So even though these helices don't have to run perfectly perpendicular, they can run diagonally. More often than not, there's going to be fairly low variability in how many amino acids are found in a hydrophobic block of primary structure as defining a transmembrane region of a protein. Oh, and that's what we just talked about. So this is just another example of hydropathy plots if you are doing an analysis of a primary structure. Now, one of the things to make this a little bit more convoluted is what makes this particular protein function as elegantly as it does isn't just the structure of the protein, it's the way those transmembrane domains are interacting with their environment. And their environment isn't just the aqueous system on top, the aqueous system on the bottom. It's also the lipid environment. And what we know now is that the lipid environment is highly variable, correct? So by making minor tuning to the biochemical composition of the lipids, or the lipid composition which surrounds these proteins that are in the transmembrane, integrated into the membrane, you are tuning the function of that protein, because these proteins' function requires on their structure, and their structure is going to be defined by how they're interacting with their environment. So the interaction of proteins and lipids within a membrane is spectacularly complex and nuanced, and it's also horribly understudied, because looking at lipid dynamics real time is incredibly challenging as a scientific question. But moving back down the list, I just mentioned how there's a fairly standard thickness for a membrane, but not, and most helices span it near perpendicular. But that's not a given necessarily. You can see from this particular image that it's okay for these regions, helical regions of hydrophobic amino acids, to run at an angle through the lipid environment. There's no reason not to. And some of the times they snorkel, or they'll run parallel to the membrane for a chunk of time. They'll go up, they'll go down. And that's not the norm, but it's also not atypical. So if you were to encounter a question evaluating the likelihood that a particular primary sequence is an integral membrane protein or not, and you saw one section which is particularly long, that doesn't preclude it from being an integral membrane protein, because it can just run diagonally through the membrane. These are just a couple pictures that I wanted to include because they are good indications of some severely tilted helices in combination with some relatively perpendicular helices. And sometimes these tilted helices, like in image B, work with each other in a sequence forming a barrel type structure. And sometimes the ability of these helices to move from a more perpendicular to a more angled structure is important for regulation, because if you have a pore that you need to open or close as part of regulation, being able to move these structures from side to side a little bit to close something off or open it up is really structurally important as part of its regulation. Speaking of barrels, here is one of the classic motifs, the beta barrel. So these are just a whole bunch of variations on the theme with varying degrees of complexity, but what you see at the core is that whereas we had helical structures before, which are alpha barrels, let me run back up, these are alpha helices, which are forming barrel structures or pore type structures. Here we have beta sheets, which are wrapping around in their saddle type structure, full circle to form a beta barrel. And these barrels can have large regulatory regions like we see in the structure of C and F, or they can be relatively simple like we see in A. But at the core, we all have common motifs which have either alpha barrel or beta barrel type structures, and then the details are in how the structures knit with one another and the regulatory regions of the protein on the inside, extracellular, intracellular components of them. This is a particularly complex structure, which is just fun to see, but you can see that we have this monomeric structure on the far right, which can interact with its multiple replicates of itself to come together, forming these very elaborate pore type structures. So this is a transmembrane barrel called WISA. But obviously this is just to demonstrate how complex the structures can be and how elegant they can be when you have single structures that work as, let's see how many we have here. We've got 1, 2, 3, 4, 5, 6, 2, 3, 4, 5, 6, 7, 8, I believe, of these monomers working in concert to form these larger macrostructures. So, moving right along, keeping the momentum going, the variation on the theme that I wanted to add was the lipid-anchored membrane protein. And the lipid anchoring system doesn't attach the protein to the membrane via ionic interactions or hydrogen bonding. Instead, it has a lipophilic region which inserts itself, or it has a section of terpene or terpenoid which can elegantly insert itself. It's really common for terpene compounds to insert themselves or integrate themselves into membranes. And for the sake of brevity, I'm just going to allude to the fact that it's very common for ingested terpenes to modify or regulate biological events via modifying the biochemistry of lipid membranes by integrating into those membranes. And if you can catch me at some time to talk about that, I'd love to, because it's really the focus of my research right now. But we just don't have time to go down that rabbit hole in this lecture. So if you're interested in it, catch me when COVID is over, and we'll have a chat. So let's get into the organization of membranes just briefly. We talked about the fundamental ideas of the model of how membranes function, but how are they organized? Well, it's really important to remember that membranes are both asymmetric and heterogeneous structures. So the distribution of proteins across the two-dimensional surface and the distribution of the lipids across the two-dimensional surface are asymmetric. Additionally, the distribution of the lipid composition from the outside leaflet to the inside leaflet is asymmetric, and that's essential for defining its curvature as well as defining the biochemical events which are taking place in the proteins housed within those membranes. Additionally, you don't see an even distribution of proteins across a membrane. For proteins that are held within membranes, you're usually going to see them, depending on their function and regulation, will be slightly dispersed, but then they'll be pulled together tightly so that they can work in unison with one another as multi-protein complexes to accomplish their biochemical transformations more efficiently. Additionally, these multi-protein complexes will often need a structure called a lipid raft, which is a lipid microdomain which typically is rich in cholesterol. So we'll talk about that in a little bit, but you'll get an aggregation of cholesterol and gangliosides and other components which are characteristic of membrane of detergent-resistant subsections of the lipids, and that these regions or rafts will then allow for the proteins to aggregate efficiently because you've changed the biochemical nature of the lipids, which allows for the proteins which are housed within them to react with each other differently. And once they associate with one another, there's the ability to regulate their function via them interacting with cytosolic proteins, which can be recruited to those constructs, multi-protein constructs at the membrane. So you see very specific things happening in terms of organization of proteins within the membrane, which allows for this really elegant metabolic function that we take for granted. So we're talking about asymmetric structure with the inside and outside of the membranes. In the outer leaflets, you see higher ratios of sphingomyelin and phosphatidylcholine, but lower ratios of the other lipids. But then on the inner leaflet, you're going to see more phosphatidylserine, phosphatidylethanolamine, etc. And you can also see it by percent breakdown of outer and inner leaflets in the B section here. But this is particularly important. And now we're going to have to get to this in a minute, but maintaining this asymmetry takes energy. It's very deliberate. This doesn't happen spontaneously because lipids can flip from one side of a leaflet to another. It happens rarely, but it happens often enough where things will tend towards an equilibrium, right? So if you have high concentration of one lipid on one side and high concentrations of lipids on another side, there's a tendency for them to equilibrate so that they're equally concentrated on both sides of inner and outer leaflets. So maintaining this is a very deliberate biological event which we invest energy in because of its function. I probably should edit this slide out because it's to make the point that we've been hammering on already, but these are dynamic processes that we're talking about, and lipids and proteins undergo enormous amount of movement within a membrane, and that these movements are essential for cellular function. The membrane is not rigid, it is highly dynamic, it's highly variable, and it's highly subject to regulation, which allows it to perform its functions that we talked about in the very first slides, which are very diverse and essential for life. So this is a really nice picture which shows some of the types of motion of the lipids can have within a membrane and how it can cause undulation. You can have lipids rotating, they can protrude, they can compact and compress, they can diffuse laterally from one point in a membrane to another by sliding around, just like if you're packed in a subway and you're kind of like wiggling through shoulder to shoulder to get, I mean, that was pre-COVID, but here we are, like latching to these analogies. And they can also occasionally flip-flop where the outer membrane lipid will move to the opposite side and vice versa, just by tunneling in and then popping out the other side. Just like the lipids, and I've been talking about this, so there's a little bit of redundancy, but here we are. The proteins which are housed within membranes are mobile, and this is essential for the regulation and their ability to work in unison together. And part of the regulation is to be able to separate them from one another and then to bring them together so that they can efficiently work in concert. And part of the thing that makes this work is the cytoskeletal component of the membrane. So in addition to being lipids, having lipid components, there's a cytoskeletal component as well. And this really sets up a lattice of cytoskeleton which fences in or semi-contains the motion of things that need to interact with one another. So they don't have carte blanche to move all about the place. They're kind of regularly contained within a region of the membrane so that they're available to be recruited to one another fairly efficiently. I talked about how you can see a lipid component move from the outer leaflet to the inner leaflet, or the inner leaflet to the outer leaflet. And this is going to happen spontaneously to try to reach an equilibrium, which we don't want to happen, because the asymmetry is essential for biological function. So it takes energy to maintain it, and the way this is maintained is through, interestingly named, Flippases and Floppases. And the Flippase and Floppase enzymes actually help maintain this asymmetry in the distribution of lipids across the outer and inner leaflets. These do require ATP, hydrolysis of ATP, to provide energy to move against the natural tendency to reach an equilibrium. And they fight this asymmetry. So ATP-dependent Flippases, or Flippases, you don't have to call them ATP-dependent necessarily, move phosphatidylserine and occasionally phosphatidylethynolamine from the outer leaflet to the inner leaflet. The Floppase moves the more amphiphilic lipids like cholesterols and phosphatidylcholine and sphingomyelin from the inner leaflet back to the outer leaflet where they belong. And then there's a third enzyme, the Scramblases, which is ATP independent but is activated by calcium, which just scrambles everything up. It randomizes lipid composition across a membrane. And you might wonder, why are we doing that? We spent all this energy creating this asymmetry. Why on earth would we do that? Well, there's times where we want to cause a rapid instability to a region of the lipid membrane. A great example would be if we were to try to insert a new protein which has been recruited maybe via a vesicle and is now going to dock and insert into the membrane, the action of something like a scramblase or sphingomyelinase will destabilize the biophysical environment of the membrane in that region, which will help allow, as everything's reassembling and reasserting itself, allow for that protein to more elegantly be inserted. So, Flippases, Flippases, Floppases, and Scramblases. And this is just a diagram showing the action of flipping and flopping and scrambling, which you can linger on by pausing if you want to give it a little bit of time. Now, I keep referring to membrane biophysics. I took an entire class in membrane biophysics in grad school, and this is a really, really short summary that hopefully we'll get through. The idea of membrane biophysics is to look at the different types of organizations that occur within lipid membranes at different temperatures, and seeing how different lipid compositions impact the relative rigidity or flexibility or fluidity of a membrane. So at low temperatures, bilayers become highly ordered and enter what we call a gel phase. And when you classically think of a lipid bilayer, you think of, you got your polar head groups and you got your nonpolar tails, and the tails are always drawn perfectly parallel to one another and they're rigid and straight. That is really a good visual representation of the gel phase. And additionally, what you're seeing in the gel phase isn't what happens under biological conditions. You're seeing what would happen in a refrigerator. So a great example of that would be, you take olive oil, you take it off the counter, and it's viscous but liquid, and you put it in the refrigerator, and at that lower temperature, it gels. It becomes rigid, similarly to margarine, perhaps. The same basic phenomenon is happening to those lipids in the olive oil under cool temperatures is what's causing the gel phase to form. We have these acyl chains that are nearly perpendicular to the membrane plane and relatively rigid. They're seeking, just like ice forms this regular crystalline pattern as it gets colder, you're forming this gel phase where, as you lose energy, you lose the mobility of those lipid groups, and they start to align with one another, and those weak intermolecular forces really dictate the regular ordered structure of the membrane. This state of the gel phase is also called the solid ordered state. And the temperature at which this transition to the gel phase takes place is really an indication of its inherent tendency towards rigidity versus its inherent tendency towards fluidity. On the opposite side of that, at higher temperatures, these acyl chains now have a lot more kinetic energy available, thermal energy available to them, so they have more ability to wiggle and move, and they're not going to get locked into this gel phase. And under these warmer temperatures, they're going to be a lot more mobile, they're going to see rotation, you're going to see translocation, and this is the liquid crystalline phase, or liquid disordered state. This is where you're going to see things happening a lot more dynamically, like they do under biological conditions. And you also see under this circumstance a decrease in the thickness of the bilayer, because these, now that you have the mobility of the ends of these lipids and they're not so perfectly aligned, they're going to slide over one another and overlap, like kind of your fingers overlapping as you cross your hands. That's going to give you the ability to compress the membrane a little bit, and that gives you a slightly smaller thickness to your membrane under normal biological conditions than you would see under cooler conditions. And the transition between these two states gives you the transition temperature. The temperature at which you see the transition is characteristic of the lipid composition. Now, it's really common when people study these to make these lipid bilayers out of 100% pure lipids that are well defined. and you get these really tight transitions in temperature. Like when you do a melting point in OChem and you have a pure compound, you get a really nice, clean melting point that's reproducible. But if you have a contaminated, let's say it's only 80% pure, but you have 20% contaminate, you're going to have a different melting point, and you're going to see a broader, more stretched out melting type characteristic. The exact same thing is true here. When you're seen with these really clean transitions between your liquid state and your gel state, that's because of the purity of that artificial lipid composition membrane. When you have a biological system, it has a lot more diversity, and you're going to have a very, very, very broad transition. And that's important for biological function if you think of it, because you don't want a membrane to go from fluid to rigid instantly. You want to have some buffering of that transition. So this would be an example of a very sharp transition between your liquid state and your gel state, and you can see, these are great diagram actually, because you can see how much more space you have between the lipid components in the liquid crystal state, and you can see how much more mobility you have in the ends of the lipids and how much more activity there is with them interacting. Membranes do undergo phase transitions, which are broadly defined by the lipid composition of those membranes. This is the specific transition temperature for specific lipids, which are often within membranes. You can see how variable they are, but once again, the whole of how the membrane undergoes its phase transition is going to be defined by these, but it's not algorithmic necessarily, and it's not neat and tidy like the image that we showed in the last slide. Now, lipid rafts. I alluded to lipid rafts in a previous slide talking about organization of proteins, and that's really what they are. So what we found is that, and this is very, very well documented at this point, is there's a microdomain that's often present in lipid membranes. And this microdomain is typically cholesterol rich, and it's typically resistant to detergents. So if you're trying to break up a lipid with a fairly strong detergent, there will be regions which resist separating in that detergent. And these microdomains were originally called lipid rafts. And that's kind of an old phrase. The notion of the raft was this visualization that you had this region of cholesterol-rich lipids and sphingolipids, which moved about like the sea of the rest of the membrane as if it was a raft in a sea of other lipids. And that's not the most accurate determination of it, because they can rapidly be separated and then formed through signaling, particularly inflammatory signaling systems. But they're typically called lipid microdomains now. But lipid raft, lipid microdomain, regardless, I'll take either, because I'm still in the habit of calling them rafts. I'm not one of the cool kids who's changed the lingo yet, even though I'll probably be forced to by going to a lipid conference soon. So that's what these are. Now, how these are essential is that they're really important for providing a specific platform for multi-protein subunits to come together, coalesce, and get you these functional multi-protein complexes. They're really, really important for the regulation of signaling. They're really important for the regulation of immune response. They're important for the regulation of redox stress. And, particularly appropriate for right now, lipid rafts are actually also targeted by a number of viruses. So viruses don't just attack any component of a cell. They target specific components of the cell. And one of the things that's very common is for viruses to attack or lipid rafts. HIV is an example. I'm not a virologist, but it's one of the things. So there's a variety of treatments associated with inhibiting lipid raft formation, which are shown to have antiviral qualities. And this is just a visual of how membrane raft or microdomain can form through aggregation of cholesterols and sphingolipids as they accumulate. And this is a little bit cartoonish, actually, because they do it fairly quickly, very quickly, and which is essential for them facilitating being a platform for protein function. Now, I talked about how proteins can separate and lipids can separate from one another and then be pulled back together based on biological need under specific signaling events. I also talked about how there's a kind of a fencing structure with the cytoskeleton which prevents these components from migrating so far apart that they can't coalesce rapidly. And if you fluorescently tag a lipid, you see is it move around through lateral diffusion within a defined region for a block of time, and then it'll hop and work within a defined region for a while, and then it'll hop into a new region. And this is called hop diffusion. And what you're seeing is lipids hop across these cytoskeletal structures. So they're not perfect barriers, but they definitely keep the lipid within a zone for a while, and they can laterally hop through this hop diffusion and spend some time within a new region, then it'll go someplace else. Now, proteins do not do that. Proteins do not hop across the cytoskeleton. So even though you have this little bit of extra mobility with the lipids moving about the cytoskeletal structure, proteins don't do that. They have to be pulled out and reinserted someplace else. And this is just a two-dimensional or a three-dimensional view of the cytoskeletal fence and how this hop diffusion can take place and how it can impact the lipid composition of a lipid membrane. So, moving on to membrane curvature. Obviously, the asymmetry of the lipid composition drives curvature, but there's a variety of other things that impart curvature artificially. A lot of cells and organelles have specific shapes that are essential for their function and regulation. And there's a lot of different ways that curvature can be regulated. So there's the unique lipid geometries and how they play in with each other. So changing lipid composition can change curvature or destabilize curvature so that it can be rapidly changed. But there's also ways you can physically force the membrane to curve by making it interact with different proteins on the surfaces, or by inserting integral membrane proteins which have conical shapes, which can act as a wedge to increase curvature as well. So here's a few diagrams of this. You can see that changing the lipid composition so that you have a different tail structure or head group structure, like what we're seeing in A, can change the way lipid membrane curves. You can see in case B, a conical membrane protein, which is a transmembrane protein, which is causing a wedge-like curvature effect. Or you can see a integral membrane protein, which is not transmembrane, non-transmembranous, which is only inserted in one side, which is driving curvature. And then we also have scaffold proteins, and scaffold proteins interact with the surface of the protein, causing a physical, like grabbing it from the surface and just causing it to curve to a specific dimension. And then lastly, there's cytoskeletal effects, which can be used to cause specific curvatures of high degrees of curvature when you're making vesicles and things of that nature, or undergoing reproduction. Now, this is just a picture of a bar domain of one of these scaffold proteins. So in D, we saw a very generic vision of what a scaffold protein looked like. This is a much more accurate representation of a scaffold protein looks like, and how these domains can interact with the polar head groups of the lipids to cause very specific curvatures along the surface of the cell. Some of these proteins can also cause the formation of vesicles. So some of these include caveolins and caveoli, and they are proteins which physically interact along the surface of the membrane to pull the membrane into a vesicle, and then at some point the vesicle is pinched off and liberated, and the membrane at the ends of that reattach with one another. And I think we've got, generically, this is an indication of how this happens and how a vesicle may contain a chemical signaling compound, and then how that vesicle can then integrate with a membranous structure across, say, synapse or something along those lines. And here's a more detailed look at how snare proteins grab a vesicle. Stepwise, 1, 2, 3, 4, 5, 6. In 1, you see the proteins recognize each other. They lock down. It's just like docking a spaceship in Star Trek. And once you get lipid-to-lipid contact, in step 3, the membrane components start to interact with one another, bridging into one another, reformulating, because you no longer have this aqueous system on both sides. So there's a reorganization inherent. The vesicle starts to open up. This is facilitated by the proteins, and then they release the contents of the vesicle to the opposite side of the new membrane. So this is becoming a long lecture because there's so much content, but we gotta press on. So how do we get transport? There's a couple types of transport we just have to discuss. Transport's obviously essential for life because we have this barrier that keeps the inside of the cell inside, the outside of the cell on the outside, but we got to get things into the cell and we got to get things out of the cell. That's the gist of transport. So that can be movement of polar things, water-soluble things, water-insoluble things, ions with charge, things that are lipophilic. There's a whole lot of complexities to getting things from one side of the membrane to the other. And all this happens via, except for in pure diffusion, via transport proteins, which are integral membrane proteins. So start with simple one, passive diffusion. You don't need the protein for that because things just passively diffuse from a region of high concentration to a region of lower concentration across the membrane. Nicotine, fantastic at passive diffusion. It goes through membranes like gangbusters, including the membranes in your brain. So nicotine's a fantastic example of a drug which can rapidly diffuse from regions of high concentration to low concentration. However, that's not really regulated transport, right? What we're really most concerned is passive diffusion. It's other forms of transport where we can have control over what's in and what's out of our cellular environments. So, here's passive diffusion again. Moving on, because y'all passed chemistry already, at least some of it. Oh, here's a twist on passive diffusion though. Here we have high concentration on side 1, low concentration on side 2, and the motive force dictating the speed at which things diffuse from the high concentration to the low concentration is entirely defined by the delta in concentrations between the two. Now, if you're dealing with charged species, you have to look at both the concentration of the solutes on one side versus the other side, but you also have motive forces defined by potential and charge. So if you have a negatively charged component which can cross the membrane, which is not easy to do necessarily because it's hard to put a charged compound through a membrane. They're not typically transmembrane capable on their own. If there is a positive charge, net positive charge on the opposite side, that is going to facilitate the pull of that negative charge to the other side. It'll have more electromotive force to accomplish that passive diffusion event, and will be therefore much more likely to occur than if you don't have a net positive charge on one side. Why is this important? Well, when you're talking about mitochondrial membranes, we accumulate proton gradients frequently in a lot of different biological scenarios. So it's more likely when you have a proton gradient to have this passive diffusion of anionic compounds that would normally diffuse. So moving away from simple diffusion, facilitated diffusion. Now, what facilitated diffusion is when a solute can only move in a thermodynamically favorable direction. So it still has to have a gradient which favors diffusion. But when you're looking at simple diffusion, as you reach towards equilibrium, the motive force for that diffusion decreases significantly, and the rate of diffusion changes significantly. With facilitated diffusion, proteins facilitate that transport, which increases the rate of transport. So even though it's already favorable, the proteins give it a little bit of a kick and really increase the rate, which is really important because when you need something in or something out through a gate, doing it quickly so you have rapid change so you can get that biological effect addressed is important. So this is just a picture which shows you the difference between facilitated and passive diffusion. Passive diffusion, much slower, and is linearly related to the concentration of the substrate. Facilitated diffusion can be quite rapid at low concentrations of substrate, which is a good thing to have because we don't necessarily have high concentrations of substrates or high deltas in concentrations across membranes to facilitate these processes. But you still see a saturation kinetic, whereas at some point as you're increasing your level of your substrate, you're saturating the system and it's going to slow down. Now, when you think of a gate, when you think of a pore or a channel, an ion channel, typically you think of a single channel. And what we're looking at here on the far left, you've got your simple magnesium and sodium channels, and potassium as well. And these are all single pore channels. What we're looking at also is number of subunits which facilitate the formation of that channel. So on the first one from the left, you have a magnesium channel, but it has 2 subunits. Next to it, you have a sodium channel, which has 3 subunits. Next to that, you have a sodium potassium glutamate channel, which has 4 subunits. Then you have 5 subunits. Moving even further to right, you can now see some proteins have multiple channels, with each subunit having a channel. And you can have channels which have 2 pores, 3 pores, or 4 pores as well. So these multimeric assemblies have a lot of diversity in how they function and how they come together and be regulated. And we're almost at the end because I'm not going to go into the huge diversity of channels or pores. It's really, I think you guys have probably been exposed to quite a bit of it in your biology classes, and there's a beautiful assortment of examples in the book. This is just a simple visualization of potassium channel, which gives you high selectivity and high conduction for potassium specifically. And you have 4 identical subunits, so you only need 1 gene to make this protein. It's relatively small. You make 4 of them, they come together and make this channel with their neat transmembrane alpha helical sections. And they give you this really nice central pore, which perfectly facilitates the undulation of potassium from one side to another without investment of ATP. Now, this is a broken down visualization of how the different subunits interact as it's facilitating the movement of the potassium from one side to the other. And here's a visualization of how this particular channel can be regulated, where on the left side it's in its closed state, and on the right side it's in an open state. In this particular case, this is regulated by a pH, where it's closed at neutral pH, but open at low pH. And so the pH effects that we've been talking about in preceding chapters on how pH can change the orientation of a protein still holds for transmembrane proteins because of how the surface components interact with its aqueous environment. So this can be gated on and off by pH. It can also be gated by, in this case, potassium channel, by voltage, which is essential if you're doing voltage gating channels for, say, neuronal function. Classic example. The potential across the membrane can also dictate conformational changes in the protein so that they open and close based on the specific voltage across the membrane. I started to include some of these extra last figures just because I think there's some value in seeing how spectacularly elegant these structures are as basically tiny biological machines which perfectly facilitate transport and perfectly demonstrate nuanced regulation from a variety of inputs and outputs. I think this is really the foundation of metabolism discussions that we have next semester. And when you see this, you can see how particularly dialed all of these structures are for their specific purpose. You can see how, in this case, this particular pentamagnesium channel is incredibly complex. It has the ability through its large structures on the cytosolic side to interact with the cytosolic environment to finely tune the function of the channel. And here are just a couple more crystal structures which indicate variations on themes with different numbers of subunits and different numbers of pores. In each of these subunits have a unique pore as well. And I guess this is chloride, water, glycerol, and ammonia flow through single subunit pores. Whew. Let me skip ahead here because I don't know what I'm looking at. 57 slides. Oh, we're right there. We're right there. 3 more slides and we're done. Getting out of here in under an hour and 15 minutes. Just like a normal lecture, but it feels longer because I don't get to actually talk with everybody, which is a shame. It's actually interesting. I found that it takes me less than half the time to talk through slides like this when I'm just talking into a computer. If I was doing this lecture in a classroom, I would allocate at least 2 classes for this much content, potentially 2.5, because so much of the classroom time involves clarifications, stories, addressing questions, addressing more questions. It's really challenging to get that real-time Q&A and interaction, and I also can tell, usually by people's faces, if something needs more energy, more time invested in it versus doing these online lectures. But I really hope, but you guys have been doing an amazing job on the take-home tests as a group. Really impressed with the amount of thought you've put into them, the amount of energy you put into sorting out all this information very quickly, integrating it with other things you've learned as part of this being a capstone class, and providing, for the most part, very insightful, thoughtful answers to the questions I've asked you. So thank you. This is a hard time to be doing higher education with COVID, and I wish we were face-to-face for these 5 weeks, but everything considered, I think you guys are doing an amazing job. So last 3 slides. Energy. So we talked about facilitated diffusion. Well, sometimes you don't have a gradient to work with, and when you don't have a gradient to work with to facilitate that diffusion, you have to put energy in. If you have to move some compound from against a gradient, from a region of low concentration to a region of higher concentration, you've got to invest energy because that is not thermodynamically favorable. And typically what you're going to see is coupling to APT hydrolysis, or in the case of lower level organisms, using light or opposite concentration gradient to move one direction to facilitate the motion of something against that, a different gradient. So classic case is the sodium pump. It maintains an intracellular concentration of sodium which is low and a fairly high concentration, relatively high concentration of potassium. And this is really important for most organs, but it's particularly important for neuronal function and the brain's function, because we have to build up these gradients so that we have an electrochemical event that lets our neurons connect with each other, in addition to the chemical communication of sending vesicles across the synapse. So ATP hydrolysis in this particular case drives sodium out, potassium in, and generally this is what it looks like. And the book has so much more details, so much more examples, but I don't really feel that it's necessary for me to put them into these slides. So take the time, take a look at how these pumps function in a little bit more detail. And then the book also provides numerous other ATP-driven pumps, as well as discussing how they're regulated. In this particular case, the naturally occurring compound ubane inhibits the sodium-potassium pump and prevents its function, which causes cardiac arrest. I believe the class of compound ubane falls into is called the cardiolipin, but I believe so, I'm not 100%. But some of these compounds, classic toxicants, function by shutting down these specific pathways or the specific pumps, so you don't get your sodium-potassium balance that you need for your neurons firing and your brain functioning. And that is it. That's it for this lecture. The last lecture in the series will be on carbohydrates. Hopefully I'll have that up by the beginning of next week. And talk to all of you guys then.